Hello everyone, my name is Shirsha Saha and today I will be discussing about the work that was recognized by TNQ this year for the Inspiring Science Award 2025. Entitled The Molecular Mechanism of Distinct Chemokine Engagement and Functional Divergence of the Human Duffy Antigen Receptor, this work has been published in Cell last year and was conducted under the guidance of Professor Arun Kumar Shukla at the Indian Institute of Technology, Kanpur. In our lab, we work primarily with G protein coupled receptors or GPCRs. GPCRs represent the largest class of cell surface receptors comprising of more than 800 different members, and these perceive a wide array of chemically divergent stimuli ranging from odorants, photons, gustatory molecules to ions, proteins, peptides, and even lipid steroids and nucleotides. And in response, they play an important role in regulating almost all of our major physiological systems. Owing to their architecture comprising of 7 membrane spanning helices, GPCRs are alternatively also referred to as 7 TMRs or 7 transmembrane receptors. GPCRs represent a major drug target, and 40% of all commercially available FDA-approved drugs target GPCRs. And these are just a few examples of some of the drug molecules that are currently available in the market targeting GPCRs for various physiological conditions. In response to ligand binding and subsequent activation of the receptor, GPCRs couple with two primary transducers, G proteins and beta arrestin, and promote signaling downstream to both of these transducers. In response to ligand binding, a typical GPCR or a typical 7-TMR signals through both G protein as well as beta arrestin. In contrast to this, a subgroup of receptors exist which in response to binding of the ligand do not signal through G protein and signal exclusively through beta arrestin, and these receptors are referred to as the atypical 7-TMRs. In this context, the Duffy antigen receptor for chemokines or DARC represents a unique 7-TMR, downstream to which G protein or beta arrestin signaling has not really been studied in great detail. Acts as a gateway for several pathogens, including Staphylococcus aureus, which secrete a class of toxins known as the bicomponent leukotoxins, which recognize DARC expressed on the surface of target cells and use this as a point of anchorage to assemble into an octameric pore and insert into the membrane, which leads to the subsequent lysis of the cell. DARC is also recognized by Plasmodium vivax merozoites, which recognize DARC, which is expressed on the surface of the red blood cells or erythrocytes through a particular protein expressed on the surface of their merozoites known as the PVDBP, and this facilitates the entry of the merozoite into the RBCs. We first started out with measuring canonical signaling downstream of DARC. So for this, we studied 7 different chemokines, which are all known as reported ligands of DARC. And following our study, we concluded that DARC does not signal through either G protein or beta arrestin. We were therefore curious to find out how does DARC signal in the cells, and for that we undertook a phosphoproteomics analysis wherein we stimulated cells with CCL7, which is one of the chemokines that bind to DARC with the highest affinity. So following stimulation, we observe that there are several proteins which undergo an upregulation and downregulation in their phosphorylation pattern. And these proteins are involved in regulating different molecular functions and biological processes. And this study therefore showed that DARC indeed signals in the cell following stimulation by its ligand. Finally, we determined the structure of DARC in complex with CCL7 at a high resolution using cryo-electron microscopy. And our structure shed insight into how DARC binds with its cognate chemokine CCL7 and what are the potential reasons for its lack of interaction with G protein as well as beta arrestin. I hope I have been effectively able to convey our work conducted, and I thank TNQ for this recognition wholeheartedly.